So thank you guys for coming to the last lecture of the regular season at late at night here. So as Dennis said, Susan and I are going to tag team this presentation. I'll do the first half. She'll finish up with the second half. I'm going to give you a little bit of a background information about the spill and where we're at with that. Talk about 2 of the 4 experiments. Susan's going to finish up with the last 2, and then conclude with a discussion on what that means for the future of the fisheries in the Gulf of Mexico. Okay, so as you guys probably know, the Gulf of Mexico is a very highly active place for oil drilling. It supplies about 25% of the nation's oil. And of all of those wells, all those yellow dots you see there below represent a different oil well, oil rig. Of all of those, about 72% of them come from waters that are 1,000 feet deep or deeper. And in fact, there's a handful now that are actually drilling as deep as 3,000 feet at this point. With all of those wells, the Gulf produces about 500 million barrels of oil per year. And I just wanted to point out, you can kind of see they're all concentrated on this side of Florida. So any of the offshore drilling for the Florida Gulf Coast was actually banned in 1995. In 2009, there was a kind of like a resubmittal of that proposal to open back up the waters again. But after the oil spill of 2010, that got completely shut down. So there continues to be no offshore drilling in the Gulf Coast along the Florida shores as of today. Okay, so let's talk about Deepwater Horizon, as I'm sure you guys have heard about this. The spill, or the rig actually exploded on April 20th, 2010, and it's been dubbed the largest accidental marine oil spill ever in history. 11 lives were lost, and the oil itself gushed out of the well for 3 months before it was able to be capped. So during that time, that meant an estimated 210 million gallons of oil were released into the Gulf of Mexico. And to kind of put that in a spatial perspective, that's about equal to the size of Oklahoma in terms of the area that it impacted in the Gulf. So that's pretty significant. Along those lines, there are also 29,000 square miles of coastline and waterways impacted directly by the oil washing ashore in Louisiana, Alabama, Mississippi, and the most western part of the panhandle of Florida. Also, that was kind of historical for this spill. Almost 2 million gallons of the dispersant was used. I'm going to talk a little bit more about that and exactly what it does. But never has the dispersant been used at that kind of a level before. And it was applied on the sea surface and also subsea where the well was actually gushing. And that had never been done before either. So this whole thing was kind of definitely a historical disaster, and we've been learning a lot from that since then. Okay, to give you guys an idea of how this impacted or the area impacted, this is a kind of cartoon drawing of where the oil had spread within the first few days in April after the initial explosion and leakage of the well. So it's about 33 square miles at that point. Well, by mid-June, you can see how much it continued to expand because, remember, it took 3 months before the well was capped. So we had, at this point, it was still gushing out of the well. And here you can also see that at this point, by mid-June, it had started to reach some of the most western parts of the Florida state line, or coastline as well. So with this amount of spreading going on and the amount of oil that continued to come out of the well, we were all very concerned, and everybody in the Gulf Coast was concerned about where this oil was actually going to end up. So the University of South Florida did a modeling study where they released some satellite-tracked drifters within these areas to help try to maybe predict how the currents are flowing at that time of year and where the oil might go. So if you want to think of it, it's similar to how they do hurricane tracking in the sense. It's all these little lines here represent a different model of a potential track of some of that oil. Well, with it reaching this part up here by June, there was a lot of concern that that oil was going to get swept up into the loop current and then eventually make its way down through the Florida Keys, into the Florida current, and up into the Gulf Stream and out. And I'm sure you guys remember hearing that. We weren't sure for a very long time whether or not it was actually going to reach the Florida Keys. But it didn't. So this is a diagram of the actual total impact of where the oil actually did hit at different parts. It might not have all happened at the same time, but this is ultimately the path of destruction, I guess, maybe, of that oil that we saw after the spill. Okay, so let's talk a little bit about what BP did to help clean up the oil. So initially, as with most oil spills, one of the first things they did was to release these contaminant booms. It's kind of like what I almost think of like a silt collector in a sense, that it's 18 to 48 inches above below the surface, depending upon how deep the water is, and it's used to either contain oil or to protect shoreline. So they were able to put some of these booms in around areas of mangroves that are usually supply nursery habitats for a lot of the fisheries that we see. And again, we're going to talk about that quite a bit, or to help contain. Unfortunately, not all of the booms worked great. Some of them actually got washed back up to shore, actually carrying more oil to shore in certain parts, and then some just completely collapsed altogether, and it didn't help. So one of the other cleanup mitigation plans that they had was the use of this dispersant. So a dispersant actually helps to break down the oil into smaller particles, so it's not quite, it kind of helps break it up at the surface. They decided to use the brand Corexit. There's a couple different dispersants out there on the market. Corexit was the only one at that time that anybody had available in quantities that were large enough to actually tackle this size of an oil spill. So that's why, with the permission of our EPA, they went ahead and started applying this dispersant. They applied it at the sea level via planes, dropping it off right along the oil slicks. And they also did, for the first time, injections right at the well site to help break up the oil before it even made it up to the surface, or at least that was the idea. The dispersant itself is pretty fast acting and pretty effective, especially within the first 24 or 48 hours. But because it breaks apart the oil and because it's applied in such quantities, there was a little bit of a concern that it's going to be moving around and moving into areas where they might not necessarily have anticipated needing to put this dispersant. Also, the dispersant works best on oil that is new, so freshly coming out of the well, versus the oil that you might see like in tar mats or tar balls on the shoreline. So as soon as they capped the well, BP stopped using the dispersant. So it was used for about 3 months straight, pretty much, in the Gulf of Mexico. So here's just a little diagram of exactly what the dispersant does. It just basically breaks that oil down from larger globules into smaller molecules. And the idea behind that is that, number one, it won't stick to some of the animals or life or rocks or whatever it is on the surface, and it will then sink to the bottom and kind of go away, except that it doesn't really go away because then it's sinking at the bottom, and we'll talk about that a little bit. But there's not just animals that live at the surface of the ocean. There are a lot of animals that live all the way in between and down to the bottom as well. So the use of dispersants has been rather controversial, always has been, not just for this spill. But we kind of have to also ask the question, was it the right thing to do? EPA put out a report in 2010, just a few months after using the dispersant, suggesting that this dispersant actually may have done more harm than just the oil alone. Recent reports that have come out, and even some of our research that we found, is that the addition of this Corexit to the oil actually makes it more toxic to the animals. Part of that is because it's breaking up into smaller molecules, and therefore the animals are either able to digest it more or they're exposed to it more because there's more around there, depending on, for example, if it's settling to the seafloor. And sometimes that toxicity can be up to 52 times greater than just the crude oil alone. There's also suspicion that some of the oil plumes that are left underwater, where they injected the well with this dispersant, actually, it created these plumes, and therefore these plumes are kind of floating around very deep in the ocean, but they're still there. And this is something that hasn't necessarily always been seen before for oil spills, and they are linking this back to the use of the subsea application of the dispersant, or they think that might be part of the reason why. However, what if we didn't use it? Well, we might be seeing more instances of this still today. Our beaches may not start to be on their way towards recovery. You might continue to see tar mats and crude oil wash up ashore. So it's always kind of a catch-22. And we talked in the last lecture that there have been some spills where we've been doing research on looking at toxicity that they won't use a dispersant at all. They'll just try, if they can contain it offshore where it happened, then they won't even bother using the dispersant. But in this case, as you can see, the amount of area that was impacted, we had to do something else about this. Okay, so let's talk really quick about some of the potential impacts aside from the obvious oil-covered bird that I just showed you. Within the Gulf, there's several different types of habitats and ecosystems that we need to be concerned with. First of all, we have your nearshore area, your wetlands, your salt marshes, seagrass areas, where they're nursery grounds for a lot of the fishery species. A lot of the mammals feed there, birds feed there, so they're obviously very critical. It's a nearshore. Even a human impact issue could occur. Then if you move offshore a little bit, you have what they call the nearshore benthos, if you want to think of it like your oyster reef beds. So of course, the oysters themselves, you don't want to be smothered with oil, but all of the millions of macroinvertebrates that live within the oyster reefs as well and thrive in there will also negatively be impacted with any type of smothering of the oil that might hit. The photic zone, if you're talking about like the upper layers of the ocean, which is where all of your phytoplankton, zooplankton, larval animals might be. You have your top predators to be concerned with. Now they're able to move throughout this area, but they might also be moving through some of these plumes that are now submersed in the deep sea that weren't there before, or they may be feeding on some of the animals in this photic zone that might have accumulated some of these toxins. And then, of course, we have our deep benthos, or especially the deep coral reefs that we have in the Gulf of Mexico. And I think over the last 2 years, we've had some colleagues actually present some of their research that they did on some of the deep coral reefs with the oil spill impacts on that as well. So for our research project, we focused on the photic zone, on the zooplankton, phytoplankton, and some of the early life stages of important fishery species for the Gulf of Mexico. So our project was funded through the Florida Institute of Oceanography, one of the very first call for proposals that went out. We did receive our money by November, I think, of 2010. So it was actually a really quick response time from the time that the oil spill actually happened. And our research was pretty basic and straightforward in the sense that our first objective was to look at just straightforward acute toxicity. So after 96 hours, what is the toxic level of the oil, the dispersant, and the dispersed oil? So the two mixed together. We chose to work with 3 different invertebrate fishery species, the queen conch, eastern oyster, and the pink shrimp. And then we chose to also work with their planktonic food sources. So we worked with some microalgae and rotifers, which are tiny little animals that the larval shrimp and larval fish will often eat. Our second part of our study was then to look at maybe more long-term sublethal impacts. So maybe these animals were only exposed for a 24-hour, 48-hour period, and then got washed away into cleaner water. Well, how would that initial early exposure affect their larval growth all the way throughout their larval cycle, which for most of these animals was about a month long. So you're talking about a 24-hour exposure after 1 month, seeing how well they did and if it affected them or had any impact on them. And ultimately, we wanted to do all of this so that we could look at impacts on the future fishery landings for the Gulf of Mexico. Not necessarily saying we were modeling that, but taking our data and looking at what the predictions might be for the future fisheries. Okay, so I like to say this is my 5 pictures to summarize 2 years of research of methods. So to make it quick and easy, basically a lot of our experiments were set up the same way. It just kind of we just kind of did one right after another type thing. We followed the standard EPA procedures and made sure that we were running our solutions and everything the right way. In this case, we were working with 1-liter beakers. We would count out any of the animals that we need, and every 24 hours, we would take a look at those animals, count the number of dead, look at them under the microscope, see how well they're swimming, see if their shells or their exoskeleton is developing normally, and see if there's any abnormalities in their swimming behavior as well. So we did this we had to collect queen conch eggs from the wild, and we hatched them here in our facility. We cultured the oysters, the microalgae, and the rotifers here on campus, and then the shrimp we received from a hatchery just down the road a little bit. So we were kind of alternating our experiments, whatever animal we had available at that time. This is a photo here of how we had our algae set up in the algae culture room. So it's slightly different for the algae. Okay, so I'm going to talk about first our results from our planktonic food sources. So, yes, the predatory fish are important, the larval fish are important, but if they don't have any food to eat, then nobody's going to survive. So we're talking about the first level of the food chain here. So we chose to work with 2 different microalgae species. This is a photo of one of them, like really tiny, literally you have to look under a microscope to see them. Species that we culture here on campus, so we were able to use them. But they're also readily available in the Gulf as well for a lot of these animals that we were working with. And then, like I said before, we also chose to work with the rotifer. Now, rotifers, not only are they an important food source for a lot of the shrimp and larval fish in the Gulf of Mexico, but they also will ingest oil. If you can see this globule here, this isn't a photo from our experiments, but the rotifers will actually take in oil, though, into their guts. And so how that translates then into survival for a larval animal that eats them is kind of something that we were curious about. So we know from past research that had been done with different types of the algae and rotifers, different species of rotifers, that they both will accumulate oil and, more importantly, the PAHs into their body if they're exposed via the water. PAHs are polyaromatic hydrocarbons, and it's the most toxic component of oil. So a lot of times, and you guys might be familiar with this is usually like the level of PAHs is what might be reported or might be talked about or what's tested for seafood safety sometimes. We want to make sure that the PAH levels are low. With the addition of the dispersant, other people have also found that the accumulation of PAHs into these lower trophic level animals also is accelerated. So we also wanted to test that. However, both of these species can also adapt somewhat to their environment a little bit better even than some of the higher level animals can. So the rotifers themselves have been shown to adapt to kind of background levels of PAHs, especially rotifers in the Gulf of Mexico that sometimes do have oil around or they're exposed to it on a semi-regular basis, not maybe to this degree, but they're there. So they've been able to adapt. And the microalgae might show a 24-hour reduced growth rate, but after that time, they kind of pick it back up and they're normal again for the most part. So it's a lag phase. So we know that they have some ability to adapt. We were concerned because dispersed oil was present in the Gulf of Mexico for 3 months. That's the longest, I think, ever in any oil spill, that we were really concerned to see how this might impact the main food source there. So what did we find? Well, pretty much exactly on par with what other people found. That, in fact, the addition of the dispersant did make the oil more toxic to the animal, and also impacted the motility of the algae. Now, several of our larval species require algae to swim in order for them to feed on it. They won't feed dead, sinking algae. So if the algae is dying, or not necessarily dying, but just not swimming, it means they're sinking into the water column. That might mean that it's not available as a food source to some of these animals. We were also able to determine what the lethal PAH levels are for these particular species of animals. So that's now reported and on the record that people didn't have those numbers before for those particular ones. So how do we kind of interpret this? Well, we know that the dispersants increase the bioavailability of the PAHs in microalgae, meaning it's more available for the microalgae to uptake. Okay? The dispersant makes it easier for them to accumulate it. We also know that the trophic transfer of the PAHs to the next level, so from the algae to the rotifers, does actually occur. But what we don't know is whether or not then the next step would happen, from the rotifer to the shrimp to the fish to whatever might be feeding on it next. And that's something that we want to, we're looking for funding to be able to kind of take it to that next step and see in our scenarios, the concentrations we were working at that we know are relevant to what was probably in the field, how that might impact those larvae. Okay, so that was the phytoplankton. Next I'm going to talk about the other animal that we worked with, the first invertebrate species, the queen conch. Now the queen conch is a very valuable fishery species, particularly through the Caribbean. Unfortunately very overfished in a lot of areas, overexploited and whatnot. But the conch itself is actually protected somewhat under CITES, the Convention on International Trade in Endangered Species. And each individual country in the Caribbean also has some type of regulations, either closed seasons, quotas, bag limits, no scuba allowed. So each country has a fisheries management plan for the queen conch. Now, in the United States, the only place that we've ever had a queen conch fishery has been in the Florida Keys, as some of you may know that. The Keys faced a total commercial closure in 1976 due to overfishing. They shut down the commercial fishing. And then they continued that moratorium in 1986, where they then closed down all recreational fishing at that point. So it's been closed for 30 years, right? Almost 30 years now. And the fishery still hasn't really recovered so much in the Florida Keys, not enough to actually be an active, even recreational fishery. In 2012, the queen conch was petitioned by an organization to actually be listed as an endangered species on the U.S. Endangered Species Act, and we're expecting to get some type of answer in late 2013 on whether or not they're actually going to go ahead and list it. So the queen conch is valuable, it's endangered, and it's in the Florida Keys. So you might ask, well, it's not really in the Gulf of Mexico, so why did we work with it? Well, I want to point out, if you recall, this graph here, or this figure here, where they were modeling a lot of these little lines, we're kind of in agreement here of where this oil might go. So in the time that we got our funding and decided, or the call for proposals came out, we said, well, you know what, we can work with this animal, we know we can raise it in captivity. Let's go ahead and also work with the queen conch, because if it does get into the Florida Keys, it could devastate the only population that's left in Florida. So that's why we chose to continue working with queen conch. And also it kind of serves as a representative gastropod species for any other gastropods that might be in the Gulf of Mexico. So with the queen conch, we wanted to focus on the early life stages, the larval cycle, and then metamorphosis, the competent stage where the animals are settling out of the water column to start crawling around and be a benthic animal. So what we did was we broke that up basically by week. So week 1, we usually have a 2-lobed larvae. And so that was our week 1 animals. Our week 2 animals usually have these 4 lobes here. By week 3, they've got 6. And by metamorphosis, which is by 21 to 30 days, depending upon several things, they're ready to settle. And so we did a quick exposure, like a 24-hour exposure at metamorphosis to see how that impacted their ability to undergo metamorphosis. So what did we find with these? Well, the acute exposure, these 96-hour studies that we did, found something very similar to the phytoplankton in that the dispersed oil was more toxic than the crude oil alone. And it actually had a greater toxic effect on the older larvae, which was something that we hadn't seen with any of the other animals, but it's something I saw before with queen conch when I was doing some research with heavy metals, and particularly copper, that it impacted the older larvae greater than the younger larvae. We also saw that the older larvae were susceptible to lethal levels that were probably found in the Gulf 24 hours post-application of the dispersant. This is based on other oil spills because we don't actually know exactly what the concentrations were in the Gulf of Mexico, or at least that hasn't been released yet that we've been able to find. But our lethal levels were lower than what we think might have actually been out there. So if these animals were exposed for even the first 24 hours, the chances are they probably didn't make it. Now, why were the older larvae more susceptible? One of the theories that we have, and Susan will talk about this a little bit with the shrimp as well, is that they're feeding more as an older animal. The first couple days after hatch, they're using their yolk sac and they're not really eating too much. So when we were actually feeding clean algae to these animals during the 96 hours, that algae was probably accumulating some of that oil and dispersant, and therefore the animal was taking it up. So animals that are feeding more actively might have had a greater impact with the contaminants than we initially anticipated. Now, even within 24 hours or 48 hours, we were also seeing shell deformities on the animals. We were seeing swimming abnormalities where they would swim in little circles, or they would just kind of hang out at the bottom and they weren't actively swimming. Like, we kind of know what a healthy animal looks like and what a not so healthy animal looks like. So even if they weren't dying, they still weren't quite acting as they should normally be acting, which is probably going to disrupt the way that they were feeding in the wild. We also saw very low metamorphic success and delay in competence. So their larval cycle lasted longer when they were exposed to all of these contaminants as well. And so this definitely has implications for these animals as they tend to settle near shore in seagrass beds. And again, at this point, nobody was 100% sure whether or not their oil was going to reach the Florida Keys. So we had definite concerns that if it gets into the seagrass beds of the Florida Keys and the offshore reefs, that there might be a huge impact on these queen conch at that point in time. We also did a sublethal exposure where we exposed them for 24 hours at a not totally toxic level to them, which was determined from our earlier studies, and then raised them in clean water the rest of their larval cycle. And we wanted to see how they were able to recover, if they were able to recover at all. And what we saw here, where we've monitored the shell growth of the animal for the entire larval cycle compared to the control, we didn't see any differences. So their growth seemed to not necessarily be impacted, which is good news. However, what we did see for the first couple of days of development within the first week is that we saw this lag period for the animals that were exposed. So this is a control clean water animal. At this stage, it has nice 4 lobes starting to develop, very clear, very healthy, extended lobes. This is an animal the same age that had been exposed just the very first 24 hours of its life. And you can see it's a little bit behind. They're not quite there yet on developing those lobes. And that's a clear indication of some type of, obviously, sublethal impact on them. Now, the lobes are used in these veliger species, any type of larval mollusk. Their lobes are used to collect food. You can see the little cilia, well, you can kind of see the little cilia here that collect the microalgae, and they're also used to exchange oxygen. So lobe development is very critical for how healthy a larvae actually is. Now, the good news is that these guys, by the time we got through the entire larval cycle, ended up looking exactly like the controls. So it was just the initial first couple of days that they seemed to lag, and then they caught back up again. And again, this is something that I also saw when I was doing some heavy metal exposures. So I had some issues and some concerns that, again, if this oil made it to the Keys, which again, we weren't sure at that time, the combination of effects of heavy metals that I discovered in the Keys and the oil and the dispersed oil in the Keys might have very devastating consequences on them. And of course, we are also concerned about bioaccumulation. So if the animals themselves are ingesting the oil, what about all of those other species that eat them, all these little babies? These are only about 0.5 millimeter to 1 millimeter in size. A lot of things like to feed on them in the wild. So how many of those were actually going to impact the next trophic level up? And again, that's something that we're hoping to continue doing some work on here in the future. So at this point, I'm going to turn it over to Susan, who's going to talk about our oysters and our shrimp, and then also what that means for the fisheries in the Gulf. I'm trying to turn myself off.
Okay, I'm going to pick it up where Amber left off, and I'll be talking about oysters and shrimp and fisheries impacts. So we do a lot of work here with bivalves, and Chrysosseria virginica, or the eastern oyster, is a very important Gulf of Mexico fishery species. As you can see, 56% of the harvested oysters in the United States come from the Gulf of Mexico. So in addition to an economic impact, which is important in and of its own, oysters are known for the ecosystem services that they provide in an environment. They provide habitat for other bivalves, for crustaceans, for larval fish. A lot of times you can see birds in an oyster bed that are picking actually those things out as a food source. Oyster reefs are also important for nutrient recycling, for stabilization of sediment, so actually building, as you can see in the picture there, building land. And they're also known for their filtration capabilities and their ability to improve water quality. In addition, worldwide, oyster reefs have been declining for many, many years, and they're considered an imperiled marine habitat. Okay, so this is just an example of the oyster life cycle, and we worked with the younger, or the first half, the planktonic cycles. So we looked at fertilization, we worked with trochophores, we worked with the dehinge stages, and we worked with what they call the eyed larvae, or villager larvae, which are right before they set, become an adult, and undergo a benthic existence. So let's take a look at fertilization effects, the first part of embryogenesis. The first thing that we found was that fertilization was not impacted by oil alone. And how we set these experiments up is we took sperm, we took eggs, and we measured out what we needed, put it in a beaker with pre-made solutions of dispersant, dispersed oil, and oil itself. And then we followed that for 2 hours to make sure fertilization had taken place, and then we moved things to clean water. However, we did find that when we conducted those same exposures to dispersant or dispersed oil, here's the dispersant results and here's the dispersed oil, that we did have an impact in fertilization. Less fertilization took place compared to the control, which is the blue one here. We took this a step further and we exposed fertilized eggs, as you see here, to oil for 12 hours or to dispersed oil dispersant until they developed into this trochophore, this free swimming stage, the first stage of development. And for the third experiment, we took the trochophore, we exposed it for 12 hours until it developed into the D-hinge stage. And in those 2 experiments, we found that development was affected by exposures to oil as well as dispersant and dispersed oil. While we were doing that, we also looked under the microscope, as Amber had said we'd done with the conch, and we looked at the effect on normal development. So if they developed, did they develop normally? And here's some examples of abnormal development. Concavity here, mantle protrusion. Some of the times you could just see a misshapen cell, divots here. Maybe it was very elongated. Maybe it was protrusion occurred over here. So there were different types of abnormalities that you look for with oyster larvae. One of the things that we found that when we exposed the trochophore stage to oil, it didn't affect the trochophores, but it did affect development to normal D-stage larvae. However, when the trochophores were exposed to dispersant and dispersed oil, they, as well as the D-stage larvae, had abnormalities. So for the next set of experiments, we looked at older larvae. We looked at those D-stage larvae, which you just saw on the previous slide, and we also looked at the eyed larvae, the stage just before they undergo metamorphosis and settle and undergo a benthic existence. As far as survival goes, the D-stage larvae were more sensitive. They were sensitive to oil, dispersant, and dispersed oil at all these PAH levels, the most toxic component of oil. In contrast, the eyed larvae were only affected by dispersant and dispersed oil. There were no effects with the amount of oil that we used. And we used up to 1,200 milligrams per liter nominal concentration, so a pretty hefty amount. Then we looked at the swimming behavior of the D-stage larvae. And in this particular instance, we didn't look at the effect of dispersant, only of oil and dispersed oil. And one thing that we found was that swimming activity was depressed at lower levels than were needed to cause mortality. And so that can have implications on their ability to escape predation or to find food, for instance. So with oysters, just like conch, we looked at short-term 24-hour exposure to a sublethal dose of dispersed oil and whether or not that would affect survival and growth. And very similar to what we found with the conch, we saw no effects on growth, nothing significant. You can see places where it looks like, okay, well maybe the exposed are not growing quite as well, but it wasn't significant. However, we saw something kind of curious that we weren't able to explain. The other experiments that we'd done, the acute ones, as Amber said, they went for 96 hours. So we looked at them 24, 48, 72, and 96 hours. These we followed them for 3 weeks until right before they became eyed and they would set. And until about, for the first week, the first 96 hours, we didn't really see any difference in survival. But somewhere between day 4 and day 8, we noticed a drop in survival in the exposed group, and that drop continued all the way through 18 days. And We were unable to explain exactly why that occurred. It was not a water quality issue, and so we theorized that it's had something to do with a delayed reaction to exposure to dispersed oil in some form or another. So we're going to switch gears and we're going to take a look at the shrimp. Shrimp are also an important fishing industry in the Gulf of Mexico. As you can see, 68% of the shrimp that are harvested come from the Gulf of Mexico. We have 3 species that, unless you take a really good look at them, you probably wouldn't be able to tell the difference, especially not once they're on your dinner plate. But we have 3 shrimp species in the Gulf of Mexico: brown shrimp, white shrimp, and pink shrimp. And of the 5 states that make up the Gulf of Mexico fisheries, some of these shrimp are more prevalent in Texas, some in Florida, some in Louisiana. Okay, the shrimp life cycle. Shrimp start out in the open ocean, and as they get larger, they migrate into the estuary where they stay until they become adults, and then when they get ready to spawn, they move back out into the open ocean to spawn. And this is basically at this point in time where fishermen actually, go to make their catches. This spawning season, there's some overlap between the 3 species, but basically it runs from anywhere from April to September, which is at the same time at which oil was being spilled into the Gulf of Mexico, so that definitely overlapped their spawning season. These are the 4 stages of shrimp that we worked with. Shrimp have quite a few stages, actually. They have 5 Nopalea stages. We worked with stage 4. They don't really look like shrimp at this point. Then we worked with Zoea 1. There are 3 zoeal stages. Still not really looking like what you think a shrimp would look like. When they get to mysis stage, they begin to have a shrimp-like appearance, the elongated body, the cuticle. So they start to accumulate a shrimp-like tail. There are 3 mysis stages, and then they finally develop into post-larvae, which are benthic. They're on the bottom, and they look a lot like adult shrimp, just a lot smaller. So those are the 4 stages that we worked with in the experiments that we're going to take a look at. So what we found was that the dispersant, which is this particular graph over there, had the greatest impact on survival. You can see the difference in scale here. These are all in milligrams per liter. So we had a much more negative impact with the dispersant on its own. Of course, we wouldn't be using dispersant if we didn't have oil. This oil had the least impact, and then dispersed oil was right in between. The other thing that we noted was that the zoeal stage appeared to be the most sensitive stage in all cases with all contaminants. This is actually the first feeding stage. Just like with the conch and the 2-lobe stage, with shrimp, the Nopalea stage don't eat, and they use the yolk sac as a food source. The cuticle is hard and well developed at this particular stage. The post-larvae, the older animals, as we would have expected, seem to be the least sensitive on the whole to all 3 contaminants. Then we took a look at behavior, swimming behavior, because in all of these experiments we had noted depressed and different types of swimming behavior, non-normal types of swimming behavior, and responses to light and things like that. So we did some experiments where we just exposed mysis and post-larval shrimp to either oil or dispersed oil and looked at its impact on swimming. And what we saw that with the mysis shrimp, there was decreased swimming ability and decreased activity at concentrations that were 2 to 4 times less than concentrations of dispersed oil and oil that were needed to cause mortality. We didn't see that with the older shrimp, the post-larval shrimp. Depressed swimming ability and activity occurred concurrently with the same levels of oil and dispersed oil that caused mortality. Then we took a look, we noticed really quickly that we saw increased molting within 24 hours in our shrimp. So we took a look at the molting activity, and we saw that with the mysis shrimp, compared to those that had been exposed compared to the control to dispersed oil, had an increased amount of molting at concentrations 5 times less than necessary to cause mortality. And we didn't see the same type of response in the post-larvae compared to the control. It wasn't a nice, neat little response like we saw here. It was kind of all over the board. You could see at the higher concentrations, you could see the oil on the appendages, you could see the oil in the guts, you could see the oil on the mouth parts, and shrimp will often molt when they're stressed or to get rid of parasites or things that are on them, on their, quote, cuticles or skin. And we think that that was what was going on here. It was a stress response and maybe a way for them to try to get rid of the oil that was on the appendages. Then, just like with the conch and the oysters, we exposed the Nopalea stage, the first stage of shrimp, for 24 hours to a sublethal amount of dispersed oil. We followed it through for a month, and we looked at differences in growth and differences in development. And again, as with the others, we didn't see any significant difference in growth. Again, there looked like there are some points when maybe there was a difference, but it wasn't significant. We did see a difference in development from the Z2 to the M2 stage in shrimp, very much like we saw a lag in development in the conch from the 2-lobe to the 4-lobe stage, and that with the ones that were exposed, they were a little bit behind, they lagged a little bit behind, but by the time the survivors had all reached post-larval stage at 11 days, both groups reached the post-larval stage at about the same time, and so that lag was short-lived. So, what does this mean from a fisheries perspective? One thing that we need to realize is that oil and fisheries are both big businesses in the Gulf of Mexico, as is tourism. So it's all kind of intertwined, especially in the state of Louisiana, where you'll have one family member who might work for the oil industry, one that might work as a fisherman, and many of the fishermen, fishing isn't done year-round. There are seasons where you fish for different things, and so they may work for an oil company or some other business, tourism or something in that off-season. And so there's kind of that conflict of interest, and in not wanting to really, go after the oil companies, because that provides income for their families as well. And in fact, during the oil spill, BP hired a lot of fishermen to use their boats to move those containment booms around, because they were closed they'd closed so much of the fishing area. So it's kind of hard to judge recovery because it's different in different locations and each spill is unique. But we can go back and we can look at some recovery stories that didn't work out so well in previous spills. For instance, way back in 1979 in the Stock 1 spill, it took over 30 years for the oyster fishery to recover. With the Exxon Valdez spill, the mussel populations took quite a long time to recover. They did eventually, but you can see from 89 to 2004, quite a long time for them to go back to pre-spill levels. And the herring population still hasn't recovered. With the Sea Empress and the oil spill with that tanker in 1996 in the UK, sea bass recruitment and populations were affected for a time after the spill. And then in 2002, with the Prestige oil spill in Spain, the fisheries similar to the Gulf of Mexico were closed up to 9 months, and it took approximately 3 years for the mussel population to recover. So recovery can take place depending on what the species is and a lot of other factors that come into play, environmental factors that are hard to tease out. The fisheries, certain fisheries may bounce back rather quickly, others may take a long time to recover if they ever recover. So let's take a look at what we know so far about the impact of the Deepwater Horizon spill on the oyster industry. In 2010, the oyster harvest was the lowest it had been in 44 years. And in some areas, oyster fishermen who had been previously harvesting up to 80 sacks of oysters a day went down to no sacks of oysters a day. There were multiple reasons for this. One, the oil spill reduced the fishing season. Remember, it was closed. Now, in some ways, that was good because it lessened the fishing pressure that they would normally have had. So even though animals were being impacted by the oil, there was no fishing pressure impact. To keep the oil from spreading to western Louisiana, fresh water was released from the Mississippi to keep the oil on the eastern side of the Mississippi. And what those freshwater diversions had the effect of doing is they killed all the oyster beds in the western Mississippi Sound. And it's not really clear, how successful they really were in keeping the oil from moving further west or not. There was also a program where oyster areas where there was no effect with oil, otherwise it hadn't reached the shores and hadn't smothered the oysters, where a certain amount of those oysters were taken and they were planted in areas that had been exposed to oil in order to encourage those oyster populations, or maybe where freshwater diversions had taken place. And what that actually did in hindsight was just deplete the oyster populations in the areas that were taken from, and didn't really help those areas where oil had impacted. So in 2012, there's demand for oysters from the Gulf of Mexico is well below what it was pre-spill, due a lot to public misconceptions, and they're a very limited season, and oyster harvesting has not returned to what it was pre-spill. Here's a kind of an example of this, looking at both all 5 Gulf of Mexico states and the state of Mississippi. So what you can see is 2000, from 2000 to 2004, how oyster harvesting went, average of all 5 states. Here's where Hurricane Katrina occurred, so you can see a depression for a year or two. Then you can see a bit of a bounce back, not quite to what it was, but a bit of a bounce back, and then you have the oil spill. And so this is what where we're at this moment. So it hasn't bounced back to where it was even after Katrina. This is numbers from the state of Mississippi. I couldn't find them for all 5 Gulf of Mexico states, but you see it follows the same pattern, even though it looks a lot more drastic. You have up to Katrina, a big dip. Katrina, a recovery after a couple of years, not quite back to what it was, but it was getting there. And then, of course, you had the oil spill here in 2010 and the 2011 numbers. Okay, let's take a look at potential impacts in the shrimp industry. So in 2012, the landings of brown shrimp are 2/3 what they were prior to 2010. In 2009, before the spill, the average landing of brown shrimp for shrimp fishermen was about 8,000 pounds in 5 days. Post-spill in 2012 was at about 800 pounds in 7 days. So a fisherman either has to be out there a lot longer, or they just have to deal with the fact that they have a lot less shrimp out there, and they're going to make a lot less money trying to be just a shrimp fisherman. White shrimp have really borne the brunt of this, and fishermen out in the Gulf of Mexico that typically, like I said, they'll fish for any type of shrimp out there, say that white shrimp are virtually non-existent. They can't even really find them out in the Gulf of Mexico. So let's take a look at Gulf-wide commercial shrimp landings. And what this is an example of, for instance, in 2010, how 2010 stacks up to a decade prior, okay? So if we just take a look at the green, we can see that in 2010, 69%, they were only 69% of the total of shrimp that were landed in the decade previously. That's what this means, and it's broken down to pre-Katrina and post-Katrina as well, but we're just going to focus on the green. This is for all 5 states combined. In 2011, we got a little bit better, 84%. It's kind of stayed steady for 2012. So it hasn't gotten quite back up to where it was, but it is improving. However, when we look at it on a state-by-state basis, and unfortunately I don't have any information for Louisiana, which would probably be the most interesting one, because they were the most impacted, but you can see that it varies state by state. So Texas, for instance, which was never impacted by oil, dispersed oil dispersant, has had no changes in the amount of shrimp that has been landed over the past decade. Whereas Florida, you can see that in 2010, we only had 64% of what we had had in the decade previously, 66% in 2011, and even a worse season in 2012. Alabama, there's not a lot of shoreline in Alabama, and they're 5 on the list of 5 as far as shrimp landings anyway in the Gulf of Mexico, but it doesn't seem to have impacted them. Again, we see an impact in Mississippi, but we see numbers in Mississippi are improving. So it's kind of a case-by-case, state-by-state, area-by-area scenario. Okay, so we'll give you wrap this up with the bad news, good news stuff. So bad news, as Amber had alluded to, the experiments that we did were conducted with field-relevant levels of oil that had been reported in other oil spills. We didn't use levels that are out of the ordinary and that you wouldn't see in an oil spill 24 hours after the spill. And yet we saw mortalities in all our larval animals. So what this does is the concern is that you lost a generation. You lost all the larval animals at that particular point in time. And so that's going to impact the fisheries, obviously. And it's going to impact non-fished animals as well. They never did recover all of the oil. It's thought that there is as much as 1/3 of the oil still out in the Gulf of Mexico. It was dispersed, ended up on the bottom of the seafloor where it remains. It hasn't degraded. It probably will be a long time until it completely degrades, if ever. And there are events that can cause it to come back up to the surface. For instance, Hurricane Isaac in 2012 stirred up sediment and brought oil back to the shore. The predicted impact so far from 2010 to the end of 2012 is that there's been a $23 billion negative impact on the economy of the Gulf of Mexico states. This is fisheries, industry related to fisheries, and tourism. And so that's a big whopping impact economically in these tough economic times that we have today. So the good news is BP was fined for all this, and those fines should be put to good use. in something called the Restore Act. The Restore Act allots a certain amount of funds from those fines to things like restoration projects and protection projects, so a good chunk here. Long-term monitoring of fisheries. And then there's pools of money that are given to the states themselves, where it's, I guess, kind of at their discretion. I don't really understand exactly how it works, but where they can divide it up and put it where they think it best is needed, whether that's for additional research, additional restoration projects, for recompensation for losses of wages and things like that that have been done. Another positive is that it is safe to eat the seafood that comes from the Gulf of Mexico. It has been tested extensively, and shrimp and oysters that are currently being consumed do not have any detectable levels of PAHs. Oysters and shrimp tend to have about a 2-year life cycle, 2 or 3 year life cycle at the time that they're harvested. So, you know, we're past that point. So we're now 3 years post-spill, and a couple slides ago we told you some of the worst case scenarios, you know, herring in Alaska never recovering and taking 30 years for oysters in the Gulf and Mexico to recover after the Ixtoc 1 spill. However, there are also plenty of stories out there where after a spill, after a year or two, things went back to normal. So a lot of it has to do with how much pollution an area received as far as oil contamination, how stable were the fisheries there previously, what's the recruitment into those areas, because that's really important if you're killing off a whole generation of larval animals. Is that area that received the oil, is that where your next generation comes from, or does it come from outside that area and it's carried on currents and then can be carried back into that area? And so I think that what we'll see, you've already seen recoveries, for instance, in the shrimp industry overall in the Gulf of Mexico, that you'll continue to see recovery, and overall in the next couple of years, barring any other ecological disasters, environmental disasters like hurricanes, you'll see things recover back to normal, hopefully back to the pre-Katrina and pre-oil spill things. But there will be places, coastal communities, that will never have a recovery because they were hit so hard, and recruitment of shrimp or oysters for whatever reason doesn't occur from the outside and impact those communities in a positive way. So with that, we couldn't have done this on our own. We had quite a few technicians from Harbor Branch that worked on this project, a couple of FAU students, summer intern. FIO provided the funds for this research, and not only to us, but to 3 other researchers here at Harbor Branch also received grants from FIO to do oil research. We were fortunate enough to be able to acquire some of the Macondo well oil from BP to do our studies with, and NALCO was very generous in giving us core exit for use in our experiments as well. And with that, I'm going to bring Amber back up here to help me answer any questions that you might have.